Hello everyone, welcome back to World of Pharmacology. Today we are going to look into the second part of Pharmacodynamics. In this video, we are going to see the mechanism of drug action through receptors. Many drugs act through specific regulatory macromolecules in the cell membrane or in the cytosol. These specific regulatory macromolecules are called receptors. Now let us see the definition for receptors. Receptors are defined as a macromolecule or binding site that is located on the surface or inside the effector cell that serves to recognize the signal molecule or drug and initiate a response to it, but itself it has no other function. So drug plus receptor gives a drug receptor complex, which in turn gives a response. The four different types of receptors are G-protein-coupled receptors or GPCR, Ligand-Gated Ion Channel, Enzyme-Linked Receptors, and finally receptor regulating gene expression. Let us see them in detail. First one is G-protein-coupled receptors. What are they? They are cell surface receptors that pass on the signal that they receive with the help of G-protein. These GPCR are found on all cell types but are abundant in the brain and gut. The G in the name refers to guanosine diphosphate or triphosphate. They are either stimulatory or inhibitory in action. G proteins are membrane proteins and consist of three subunits, that is alpha, beta and gamma, and hence they are called heterotrimeric. The beta and gamma subunits modulate certain effector like receptor operated K+ channel, adenyl cyclase and phospholipase C. The following figure shows the structure of a GPCR. It has 7 helix in its structure, so it is called as heptahelical receptor or serpentine receptor or metabotropic receptors. So a ligand that is binding to this GPCR causes G-protein activation, which leads to intracellular chemical events through complex process. GPCR controls cell function through three major pathways. They are through adenyl cyclase, that is CAMB pathway, phospholipase C, IP3 DAG pathway, and the third one is through channel regulation. Adenyl cyclase or CAMP pathway. CAMP is cyclic adenosine monophosphate. Activation of adenyl cyclase results in intracellular accumulation of second messenger CAMP, that is cyclic adenosine monophosphate. The CAMP acts through CAMP dependent protein kinase, which is represented as pKa. With this phosphorylates and alters this pKa phosphorylates and alters the function of many enzymes, ion channels, transporters, and structural proteins. So the effect produced is contractility or impulse generation in heart, relaxation of smooth muscles, glycogenolysis, lipolysis, hormone synthesis, etc. In addition to the above mentioned functions, cyclic adenosine monophosphate directly opens a specific type of membrane calcium channel called cyclic nucleotide gated channel CNG in the heart, brain and kidney. Inhibitory G protein inhibits adenyl cyclase and opposite response to the above are produced. Examples of receptors that regulate CAMB pathway through G protein are stimulatory G proteins are beta adrenergic receptors, glucagon, H2 histamine, D1 dopamine. Inhibitory G proteins are alpha to adrenergic receptors, M2 muscarinic, and D2 dopamine. Next one is through phospholipase C, that is IP3-DAG pathway. Activation of phospholipase C hydrolyzes the membrane phospholipid PIP2 to generate the second messenger IP3 and DAG. This IP3 mobilizes or causes the movement of calcium from intracellular organelle depot, and DAG also enhances the activation of protein kinase by this calcium. Cytosolic calcium acts through calmodulin, PKC and other effectors and mediates or modulates the contraction, secretion or transmitter release, metabolism, etc. This PLC can also be inhibited through inhibitory protein. Examples of our receptors are M1 muscarinic receptors, H1 histamine, and alpha-1 adrenergic that uses phospholipase C or IP3-DAG pathway. Through channel regulation, the activated G protein can also open or close ionic channels that are specific for calcium, potassium, or sodium without the help of any second messengers like CAMP or IP3, and can bring about hyperpolarization or depolarization or changes in intracellular calcium. The G protein GS open calcium channels in myocardium and skeletal muscles, and GI and GO open K+ channels in heart and smooth muscles, as well as close the neuronal calcium channel. So this causes physiological response like transmitter release, neuronal activity, and smooth muscle relaxation. Examples are beta-1 adrenergic in heart skeletal muscle, which increases calcium ions. Receptor is ligand-gated ion channels. Here, the drug binds directly to the receptor that is located on an ion channel without the mediation by G protein or second messenger. So here, binding of the drug to receptor takes place, which opens the ion channel. The opening of ion channel causes the flow of ion through channels, which in turn causes hyperpolarization or depolarization. This hyperpolarization or depolarization causes a tissue response. So this is how the action takes place. This type of receptors are fast acting. Examples are 5-HT3. All other 5-HT receptors are G-protein-coupled receptors except 5-HT3. Then GABA A and nicotinic cholinergic receptors are the examples of ligand-gated ion channels. This enzyme-linked receptors. This type of receptor has two sites, an extracellular site and an intracellular site. The extracellular site is the drug binding site, and the intracellular site has enzymatic activity. This extracellular site and intracellular site are connected through a single transmembrane stretch of peptide chain. The intracellular site that lack the intrinsic enzyme activity bind a JK stat on activation. So let us see how this enzyme-linked receptors work. First, the action of an intrinsic enzyme receptors. On binding of agonist to the extracellular site of enzyme-linked receptors, dimerization of the receptor takes place. This dimerization of the receptor causes activation of tyrosine protein kinase of intracellular domain, which in turn activates intracellular signaling pathways, which causes gene transcription ultimately resulting in a tissue response. And next one is the action through a JK stat. Next is the action of JAK stat kinase binding receptor. JAK is Janus kinase and stat is signal transducer and activation of transcription, which is another protein. The intracellular site of these receptors lacks intrinsic protein kinase activity. So binding of agonist to the extracellular site of enzyme-linked receptor induces dimerization of receptor, which activates intracellular domain to bind free-moving Janus kinase molecule, and JAK is activated, that is Janus kinase is activated, and which causes phosphorylation of tyrosine residues on the receptor, and this tyrosine binds to STAT, that is STAT protein, and tyrosine residue of STAT is formed. This is phosphorylated by Janus kinase, and this phosphorylated pairs of STAT dimerize and translocate to nucleus, causing regulation of gene transcription, which in turn gives a biological response. Examples are cytokines, growth hormones, etc. The fourth receptor is the receptor regulating gene expression, transcription factor. These are intracellular, that is cytoplasmic or nuclear, soluble proteins which respond to lipid-soluble chemical messengers that penetrate the cell. These receptors, after activation by hormone binding, act on the genetic material in the nucleus to initiate or inhibit the process of transcription. See the action in details. The hormone binds near the carboxy terminus of the receptor, which exposes DNA binding domain in the middle of the receptor, causes attachment of the receptor protein to the gene and facilitates their expression. Now, specific mRNA is synthesized on the template of the gene, and this mRNA moves to the ribosome. And thus directs synthesis of protein and regulate the activity of target cell. Examples are all steroidal hormones, that is glucocorticoids, mineralocorticoids, androgen, estrogen, progesterones. Vitamin D, thyroxine, vitamin A functions in this manner. Thank you for watching. We will see you in the next class. Bye.